Thank you, Emma, for having me. I just like to echo that, really the UK patient and research community is clearly very energized, and it's so exciting to see the patients, the funding agencies, and especially Rob really become one of the major global focal points for ACC research. It's just fabulous what Rob's doing, supported by all of you. We're really excited about that. So just a quick background on our foundation. It started from the patient community. This is me and my wife, Marnie, who was diagnosed with ACC in 2004 in a right parotid gland. This was the week before she went in for surgery, us and our 4 boys. They were 9, 7, 4, and 1. So it was incredibly hectic time, which I know many of you all have gone through exactly that kind of hectic time. But the point is that, this is patient driven with the urgency of the patient community. When we started it, I thought, 5 years we'll have the cure. And as Rob was saying, sadly, science moves more slowly than that. To do it right, to get all the building blocks in place and to make sustainable progress, goes slower. But we've stuck with it. And really the quick conclusion is, we have made great progress. There's plenty more to do. We don't have a cure, but we're getting closer and closer to better treatments. And it's driven by the patient community. So here, just quick overview. Our mission is very simple. We want to accelerate the development of better treatments and a cure for ACC. That's a great goal, but how do you concretely get there? The way we think you get there, the way that patients end up taking better treatments, is by developing a pipeline of clinical trials that are based on the best available science. You can read a story on the internet that says, hey, this herb cured all my ACC, and you could chase all of those down. But what we think matters most is having good science that's carried out at good centers like the Christie, where you get good hard data and patients can know that the treatments are really working. These clinical trials don't just pop up out of nowhere. You really need to build a community of researchers who are following a coordinated plan that's driven by supportive and supported patients. As I was saying, fabulous researchers like Rob, need to be supported by the whole community, and you guys are doing that, and we're trying to do that, and that's how we get to powerful new progress. A quick view of the overall global network that's been created. Certainly, patients are a big part of that at the top. We have a lot of academic institutions. ACCRF has supported about a little over 50 researchers at about 30 different institutions globally. Here I've only listed some of them, but you see it really is a global network: Australia, Germany, Korea, Netherlands, Sweden, UK, and a lot of US as well. But it really is global. You also see that private industry, at the end of the day, it's private industry that is going to actually manufacture the treatments and distribute them. And so, you have to have good relationships with industry to bring in their drugs and test them. And they're usually going to be the ones that fund the clinical trials. And we really, ACCRF has signed agreements with probably about 30 to 35 different companies. So we have great relationships with a lot of companies. And then we also likewise are very involved with the public funders and other charities. I've been on many different councils of the National Institutes of Health, the National Cancer Institute, the American Society of Clinical Oncology. My wife has been on many NCI bodies. We've also coordinated with other foundations. And I know that Cancer Research UK does a fabulous job of coordinating a salivary gland working group that Rob is one of the co-chairs of. So, it really, what's crucial is to create this whole environment of support, different folks that are really engaged and collaborating well. And we see ourselves as sort of sitting in the middle, giving money when we need to, hosting scientific meetings, hooking up researchers who obviously should be working together but may not know about each other. All those things, any way we can sort of help this environment, that's what we do. Our overall research agenda, everything starts with the patient community and ends with the patient community. Nothing gets done in research without patients. Patients have to donate their specimens, their medical records, their funds. They've got to tell people, tell researcher what matters to them, and they need to enroll in clinical trials. Then they hand it off to the research community that hopefully works their magic. But the outcome of that whole process is coming back to better therapies and outcomes for patients. So it really is a cycle. Just in the interest of time, I'm not going to go through our, sort of what we expect of the research community, but all I'll say is that this first step, which is creating the building blocks of research, that's absolutely crucial. Nothing happens in research unless you've got biobanks and registries and cell models and mouse models, all of these feed into so many of the genomic studies that that really get to tearing apart the biology of the disease. And this is why it's so important, the support that you all have given to Rob. Rob is really, he's created a biobank, a registry. He's working on the patient-derived xenograft models. These are all things that, it's great. They build the research at Manchester, but they also get shared globally, that the data gets shared globally. And I'm going to talk about a project later where Rob is involved in that, and that's absolutely crucial. And this environment, this whole ecosystem really is working very, very well. Quickly, looking back over the last 15, 16 years, the landscape of ACC research has been totally changed. It used to be that there was very little funding. There were no preclinical models. We didn't understand anything about the genomics of the disease. There were very few clinical trials, and the ones there were really made no sense. They were just sort of throwing spaghetti on the wall, seeing whether the next drug worked. Now you look at today, ACCRF, we've raised more than 20 million. There's well over 25 million that's been funded by the US National Institutes of Health and many other institutes. So the funding has really taken off over the last 15 years. I was talking about the building blocks of success for research, the biobanking, the cell lines, the animal models. That's been changed dramatically. We now have specimens, we have preclinical models, we can do the research now. From that, we've understood much better the genomics of the disease, what are the drivers of the disease. For those of you who aren't that familiar, cancer is a genetic disease, but that just means that the genes in particular cells are altered. It doesn't mean that it's inheritable. It has to be inheritable. And ACC generally is not viewed as an inheritable disease. So whenever you hear genomic or genetic, yes, in individual cells, the genes have been altered, but not in all the cells in the reproductive cells, which would make it inheritable. And so don't stress out about that. But we understand so much more about the genomics of the disease. We've screened actually probably closer to 130 different anti-cancer compounds in our mouse models. And the clinical trial landscape is much, much better. We now have some drugs that are like lenvatinib and axitinib that seem somewhat active. They're not cures. We need to do better, but we're starting to see the results of this overall amazing effort. If you look at these are quick trends and trials, really this past year we had, I think, 14 trials. Currently, I think we're about 12 or 13 that are open. A couple have closed more recently. But we expect more to open on the horizon as well. So this has just been much, much more active over time. I tell you what, just in the interest of time, I may quickly gloss over, sort of the biology of ACC. What I'll say is that what we know is that about 90 to 95% of ACCs have too much of these genes, either MIB or MIB-L1, and that is really enough to sort of get the tumor started and for them to grow slowly. What we see is that other tumors grow much more much more quickly, and that's often associated with getting additional alterations, such as in Notch1. Rob pointed out his recent paper on TP53, another really important tumor suppressor gene. Those are the types of studies we need to understand the disease with, but that's another example of something that's going to make a disease a little bit more aggressive. And so you start with MIB or MIB-L1, and then you tack on these additional alterations, and we think that's what drives more aggressive or more late stage disease. If we look at all the different clinical trials that there have been, I've categorized them sort of by different mechanisms of action, by different targets. So the most advanced are these multi-kinase inhibitors that go after VEGFR and FGFR and PDGFR, and that includes lenvatinib, apatinib, axitinib. And so we kind of know that those have some activity. And then, as Rob was saying, there are these newer trials where they're combining these multi-kinase inhibitors with immunotherapies, such as avelumab and pembrolizumab. And that's sort of the next active area, immunotherapy. We've had a lot of trials in this area, and as Rob said, it's, there've been some responses, but really not impressive, and that's why we need to combine those immunotherapies with other drugs to try and increase the activity of those drugs. We also have some drugs that we hope are going to hit MIB. We have some that we also know are going after Notch. These trials are open. There are these other targets, MDM2, PRMT5, PSMA, and HDAC. All of these, we've seen some preliminary activity. There's some activity of these drugs, but we're going to have to wait probably at least till the end of this year and maybe early next year before we get good data on whether some of these targets are really working out well for ACC. But overall, you can see it is a very active landscape. So that's kind of the review. Let me just go over a little bit prospectively. I want to draw your attention to a new study that is just now opening, and it's very different than all those other clinical trials I was talking about. All those other clinical trials were for patients with recurrent metastatic disease, and so a lot of those companies are getting closer to going to the regulatory agencies. So the US FDA or the EMA, and I blank on the UK version. It used to be within the EMA, but now no longer. So a lot of companies are going to those regulatory agencies trying to get approvals for their drugs, and you can get it based on the responses, the tumor shrinkage or extended life. Another way you can get approval is by seeing that the quality of life of patients is improved, that pain goes down, that they're able to undertake more activities. And so this is our first study where we're trying to understand, very methodically, what is the burden of ACC, just across patients generally, and then across patients with more aggressive Notch active disease. And so the idea, if we understand not only whether the tumors shrink, but also we understand whether the patients feel better, they are able to be more active, whatever it is that makes a patient's quality of life better, we can measure that with the drugs as well. And so this study is just now open in English. It will eventually open in Hebrew, Russian, Arabic, and but right now it's just in English. And if you all have an interest in it, you can email me later. You can see this is our website. If you go under the Living with ACC tab, and then there's a Take Action tab under that, will take you to study, and this is what will come up. And then this will describe the study. and there'll be a link to take a survey if you're interested in participating in the study. But we think this is an important study to not just come to better treatments, but also get them approved so that regulators understand that they're benefiting patients. Looking forward kind of over this year, actually our board is meeting with our scientific advisory board and our board of directors. We're meeting over the next month to sort of firm these up, but this is sort of preliminary research priorities for this coming year. As always, because as I mentioned before, this drug MIB is sort of what we think is the main driver of ACC, and so we have various portfolio of approaches trying to go after MIB. We've also talked about Notch. That's an important driver of disease, and we already have, as Rob mentioned, 2 different drugs in the clinic that are going after that. And we see some activity that has been reported, but again, it's not good enough. And so what we're also looking at is combinations in our preclinical mouse models with the idea of coming up with new, better clinical trials in the future. So hopefully that will read out over this coming year. We also have a horrible history of not being able to develop cell cultures in ACC. Usually in cancer research, you have cells growing in plastic dishes very, abundantly, and you can do lots of cool science on them. Sadly, in ACC, that hasn't been the case, but we have some very, very promising new approaches to developing 3-dimensional cell cultures, which would allow high throughput drug screening and maybe even genetic screening. So that's a major priority for us. And what's nice is our pre-existing mouse models, in which Rob is developing as well, feed beautifully right into that. We have you can take your human tumors that are growing in mice and then quickly develop them into cell cultures. And so if we prove out this platform, it will be very, very helpful for researchers globally to use it. We also have a project going on that's using artificial intelligence, deep neural networks, I think is what they're called. And we've got a nice consortium of researchers, including Rob, that's making available these images of tumor slides, and they're all being correlated with histology and the genomics. And the idea is, can you easily diagnose different types of ACC, perhaps even diagnose, say, a Notch mutant tumor from just looking at a slide? So it would be much quicker, much cheaper to diagnose and decide on treatments for patients using artificial intelligence. There's also an effort to look at oligometastatic disease. That's so about half of patients with ACC have surgery and radiation, and actually the disease really may be gone at that point. It may never come back. They always need to be followed because ACC recurs a lot, but maybe close to half of patients, surgery and radiation may be enough. We know that half or maybe a bit more of patients will get metastatic disease that will travel to the lungs, the liver, elsewhere. There's a new focus of a lot of cancer researchers, especially in lung cancer, but also I think prostate and some others, where what they found is that patients who have just a few metastases, 5 or less, if you treat them with ablation, so it could be radiation or radiofrequency ablation or cryoablation, or even surgery, if you get rid of the few metastatic deposits, lesions that are there, there has been in those other diseases, in non-small cell lung cancer, for example, a survival benefit from treating oligometastatic disease. And we're trying to understand, first of all, how often do ACC patients have oligometastatic disease? And secondly, if you treat them, is there a benefit? Does, at least in progression-free survival, do do patients live longer without the disease growing? Or hopefully over time, we might even see if there's a overall survival benefit. But that's another interesting new area that we're looking at. And lastly, I'll mention this PSMA, which is prostate-specific membrane antigen. And it's, as its name indicates, it's usually specific to the prostate. However, that's not really true. We know that a lot of ACC tumors, in fact, it was like 94% of ACC tumors express a fair amount of this PSMA. And what we're doing is we're trying to piggyback on the prostate cancer clinical trials that are out there. So there's already a very cool radioligand therapy trial that's opened in the Netherlands, where you get an antibody that is injected into your body. It goes throughout the body, finds where there is this PSMA, and hopefully that's an ACC tumor, and it's carrying a cargo of small radiation particles that then bring the radiation to those tumor cells and kill those cells. So that's one way of saying wherever there's this PSMA, we're going to try and bring something that will kill it. There's also, and that's already in clinical trials. There's a clinical trial in the Netherlands. We're pretty sure this year here in the US, I think at least 4 sites will open a similar clinical trial for PSMA radioligand therapy. But there's also a whole new, much earlier stage approach, which is looking at CAR T cell therapy, which is another type of immunotherapy where it looks for PSMA and teaches the immune system T cells to look for PSMA throughout the body. So that's something where it's very early stage in prostate cancer, but we're just starting to look and see if that might be something that might work in ACC as well. But that's very, very early stage. So those are just some of the research priorities, and hopefully I haven't talked too long, but that's, I'll stop there and take any questions that you all have.